Her name is pronounced C. The H is silent, so I can probably get it to stop doing autocorrect because it always says it's Jane His is here. So anyway, Jane is going to tell us about microscopy. So it's another one of the little things we can't see. Next week, if the Zoom works from Lawrence Radiation Lab in California, we will have Emily Gunger telling us about the littlest particle in our series, the antineutrino. So, Jane, take it away. Thank you for coming.
Can everyone hear me okay? All right. All right, a little bit louder. Okay. Okay, is this good? Okay, cool. Yes. All right. Hi, everyone. My name is Jane C. Thank you for having me here today. I am a third year PhD candidate in the McKenna Lab at UF. And today I'll be talking about how our lab applies cryo-EM or cryogenic electron microscopy to study parvoviruses. Perfect. All right, to begin, we're just going to talk about what a virus is. As the header said, viruses are really small, you can't see them, and they're pretty mighty, and they're also everywhere. So these are the smallest of all microbes, and they cannot replicate effectively on their own, and that's why they need hosts or organisms to hijack their cell machinery in order to replicate efficiently. They are ever present, they affect and interact with organisms really at all levels, and often they're negatively associated with disease because they're the cause of it. So because we're a structure lab, I wanted to introduce you to four main kinds of structures. So here you have helical viruses, which you can see form a helical kind of sheath. So, and an example of this is a tobacco mosaic virus. The viruses that we study actually fall under this polyhedral virus family. You can see it forms a kind of icosahedral. There are other types of viruses I'm just going to mention to let you know that the types of viruses I'm talking about aren't the only types of virus structures there are out there. So you have spherical viruses, which is like a kind of envelope. It doesn't form a geometric shape. A famous example of a spherical one would be coronavirus, which we're all very familiar with from the last few years. Lastly, you have the very complicated viruses, such as this bacteriophage, where it has something that looks like a polyhedral virus as its head, but then has the helical body or tail sheath that works to inject its genome into its host cell. So to simplify this, I can say that viruses have two main components that you have to be aware of, which is they have a genetic core, which can be made of DNA or RNA, and essentially this is kind of their blueprint for how they can make more of them once they get into their host cell. And then to protect this bit of genetic core, they have some sort of protein coat, and in our case, we call this a capsid. And then, as we said, we're focused on the kind of polyhedral type of viruses. So here's just kind of a fun fact that an icosahedral is part of the Platonic solid, would comprise of 5 geometric solids. And to be a Platonic solid, all the faces have to be identical and meet at the same 3-dimensional angles. And some fun pictures is there's only 5 of them that we know of, which include the tetrahedron, the cube, and the octahedron, as well as the dodecahedron. And our viruses form this kind of shape, which is an icosahedron, or, in the popular D&D game, the D20 dice is what you can think of. All right, so now that I've given kind of a brief introduction to what viruses are, their main components, and the kind of shape that we're looking at with our viruses, our lab specifically studies parvoviruses, which is just a family of viruses called Parvoviridae. And this family of viruses is further classified into 3 subfamilies. So here you have Parvovirinae, which is a subfamily of this family that only affects vertebrates, so ones with a backbone, such as humans. And then you have a second subfamily called Denslovirinae, which only affects invertebrates, so things without a backbone, think like crickets, bugs, shrimp. And then third, you have a third subfamily, which is cool. It was only really reorganized into this third family in 2020. And this subfamily can affect both vertebrates and invertebrates. And within each subfamily, you have something called a genus or a genre, which is just further classifications of the subfamily. So parvoviruses are single-stranded, non-enveloped DNA viruses. So here you can see inside the capsid, you have the single-stranded DNA that carries kind of all the instructions for how to build more of itself once it affects and hijacks its host cell. And then here you can see it is protected by this T=1 icosahedral symmetry, and the diameter of this is about 250 angstroms, as mentioned, can't be seen by the naked eye. Okay. And then here is just to show some of the genre that I've worked with in this lab specifically. All right, so here's to go into kind of the building block structures of a parvovirus. So in single-stranded DNA that I mentioned earlier, you have these different open reading frames or instructions that you have. VP just stands for viral protein. So to be efficient, that whole capsid isn't just one piece. It's actually made up of 60 individual viral protein pieces. And one of the single of the 60 viral proteins looks kind of like this, where you have these beta sheets that have this jelly roll motif, which you'll hear me mention often is conserved amongst all parvoviruses. And this part of the monomer is actually facing towards the inside of the capsid where the DNA is. And then you have these regions that face the external of the capsid that can be called variable region or surface loops. And this is different amongst all the parvoviruses because this is external. It changes the morphology of the capsids, and depending on those changes in the surface, you might target different hosts or different types of cells. So you go from this one monomer, and how do you make an entire capsid? And here I have a model that you guys can look if you'd like. So you have this monomer and it has to form the icosahedral symmetry axes. So if you take 5 of these, they form this little like cute channel that you can see here. And the channel is important and it has to be with all the viruses because it's speculated that's where the DNA is ejected or such. And then if you take that, you can see that on the entirety of the capsids, it makes up this region over here. Another one you can look at is the threefold protrusions, which then also appear on the capsid here as well. And I don't know if you like, but we brought some, this is like the threefold in 3D of what it would look like. And here is just the monomer, the blue part being the conserved beta region. Right. And then on top of these ones, so just to reiterate, these are kind of symmetry axes, 5-fold, 3-fold, and 2 5-fold wall or 2-fold depression that all parvoviruses will have. So you can notice the symmetry amongst all the capsids. Okay. All right, so as mentioned earlier, that's pretty much the basic building blocks of every capsid, and there's 60 of them that form together intricately to make this capsid. Here's just to show that although amongst different parvoviruses, this region here is pretty much the same if you overlay them and superpose them. There's a lot of differences in the surface loops. You can see that at least in this region 8, that some of them MVM is longer or some are shorter than others. So that's going to contribute to why their capsids look different. Yep. So here you can see that different variable regions contribute to different parts of the capsid. And by comparing them, you can kind of analyze why some maybe affect certain hosts or certain cell types. Okay. And the reason why the capsid is so important, it's part of its life cycle where in order to recognize a receptor and enter the cell depends on what the capsid looks like, because certain capsids will bind to certain receptors and then be able to attach to the cell, and then it'll be able to enter the cell using receptor-mediated endocytosis, and then it's trafficked, and then eventually it's able to leave this little endosome using this something called VP1U, which has a PALL2, which cleaves the endosome, allowing it to escape, going to the nucleus, and then therefore giving its instructions and making more of itself. So that's why capsid structure is very important. Right? And then because of the differences in surface loops, these parvoviruses can affect a really wide range of hosts. Here within just the Parvovirinae, which is the subfamily that affects vertebrates, you can see everything from dogs to minks to birds to humans, horses, snakes. So really, it's pretty impressive how much that, even though the inside is the same, just changes in the surface loops and variable region could lead to a huge wide range of hosts. And what's important to note is that parvoviruses can be pathogenic and associated with disease, but there's also some that are actually non-pathogenic and don't cause disease as well. Okay. And there's some really cool reasons why our lab studies parvoviruses, and here I'm just giving kind of a few of those. So first of all, the beneficial ones is that ones that aren't associated with human disease could actually be used for good. So there are certain class of parvoviruses known as oncolytic parvoviruses that prefer to replicate in just cancerous cells. So they could potentially be used to target cancer specifically and be a cancer treatment option. Much research and investigation is still needed in this field, but that's a pretty cool thing. Another thing that's been really extensively studied, so within the parvoviruses, there's a genre called dependoparvoviruses. And mostly because these viruses rely on another virus being there to replicate efficiently. And UF is really big for this field of adeno-associated viruses. So these are viruses that are non-pathogenic. They're extensively studied for the use of gene therapy, meaning that you can package a transgene that is beneficial and then deliver it to the host in hopes of curing a disease or such. So really cool. Another cool thing is that for the parvoviruses that actually do cause disease, knowing their structure could help us with vaccine design or antiviral drug design. And also the reason why studying structure is so important is that we want to prevent cross-species transmission where a virus is able to mutate and originally only affect one host, but mutate enough to also spread to other hosts, which is never a good thing. As shown here by the historical example, if anyone remembers puppy parvovirus, so, or canine parvovirus type 2. This first emerged around dogs in Europe around 1976, and this caused a really wide outbreak, many puppies dying from gastroenteritis and other symptoms similar. And what was really cool is that it wasn't essentially a completely new virus. It actually came from feline panleukopenia virus, which then only had these 5 to 6 changes in its capsid structure and then was able to effectively spread to canines or dogs as well. So, yep, these are just some reasons why we study viral capsid structure, which I think are pretty convincing reasons. All right, so here's just kind of historical look at how many years. So this is a pretty seasoned field with, if updated, should be 30 years of parvovirus structure biology. And you can see at the beginning, most of it here was done with X-ray crystallography. And here is a very historical McKenna Lab paper on the structure of canine parvovirus and its functional implications, which is pretty cool. So we have a long running history of working with parvovirus in our lab. And then with the emergence of cryo-electron microscopy, which is the technology my talk is focused on, you can see that over the span of time, cryo-electron microscopy has started to really dominate the field of the method of choice to studying these parvovirus structures. And the highest resolution to date we have is 1.6 angstrom, and later I'll talk what that number means to explain what is a good resolution versus what is a poor resolution. All right, so these were the advancements in cryo-EM. So X-ray crystallography has always given pretty good resolution, and the better the resolution, the more accurately you can see how the capsid looks. And here is, back in the day, the resolution is 22 angstrom, and initially using cryo-EM to study viruses was called blobology, because you would see these blobs and try to make sense of what they did. But with advancements of better detectors, as well as more powerful microscopes and better computers with Hypergator, we now have much better supercomputing power. 19/20 years later, we now have a structure that looks much clearer like this. And what's really cool is my mentor worked on this structure back in 1999 of a parvovirus. Okay. And then the reason why cryo-EM has kind of dominated over X-ray crystallography in recent years is that cryo-EM does have some advantages, which include that in X-ray crystallography, before you're able to resolve structure, you have to first get your biological protein to form a crystal. And that in itself is considered kind of a dark arts. That's very difficult to accomplish, testing many conditions. Secondly, because of the nature of how we prepare the sample for cryo-EM, which is vitrification, that arguably maintains a closer to kind of biological state. And then you can determine structures of larger complexes. You only need a relatively small amount of your sample to perform this structure determination. And secondly, as mentioned earlier, the advancements that we've made in the last 20 years have allowed us to have increasingly really good resolutions at high speed and relatively good affordability. Right, so here I kind of get into what is cryo-EM now that I've talked about that it's really important and used a lot in our field. And pretty much a kind of general way of saying it, it is snapping pictures of flash frozen things. And it allows scientists to make kind of enlightened 3D images of DNA, RNA, proteins, and in our case, viruses. And then by stringing thousands of these snapshots together, we can actually reveal the structures itself. And here's kind of a general image of you have an electron beam, and here are your samples that are flash frozen into a glossy state. And then because of this, it casts shadow on the detector here, giving us images that we then have to go into the computer to computationally resolve the 3D structure. Right, so I'm gonna go kind of step to step, kind of what those steps are. So the first thing is here we have a grid, which is often coated with carbon layer with holes. So this is what our sample is gonna go onto before it's shot by the electron beam. Here's kind of a close-up to see those small holes, and the small holes are there to kind of help suspend the sample, which is usually in liquid. Right, so here you would apply your sample to the grid. You would kind of blot the grid to remove any excess liquid. And then here is that glassy plunge freeze I was talking about. Nowadays, we have a robot do this for us. It's called the Vitribot, and that vitrifies the sample. And it's in liquid ethane, which is a negative 172 degrees Celsius. It was very cold. And then that allows you to have these kind of thin vitreous layers. So it's not quite the same as ice, because ice would probably cause damage to the grids and you'd have ice crystals, but it's like a super cold state where it is frozen, but the samples inside can still move around. All right, then you have the data collection part, where usually our lab would prepare the sample and we send it off to a facility that has the really powerful equipment in order to do this. So including Stanford SLAC is a good one, or UCLA, and I think FSU. And you'd have your electron beam go through that glossy sample that I talked about, and your samples are kind of free floating, and then your detector would then capture the shadows of that. And you can see it's actually pretty cool that because your samples are free floating, you're getting different orientations. And you can see over the years that we've gone better and better, and I'll talk about how lower number is better resolution. However, because the samples are moving around, the electron beam induces more movements, which can lead to blurry images, which is not what we want to work with. So through technology and averaging, we're able to now align them and go from a blurry data set to a motion corrected and nice data set. And here, this is example of what our capsids would look like on the detector. So you can see these little circles. And what's cool is in this one, you can see the ones with a sharp outline on the outside actually don't have DNA package. So sometimes viruses are not always perfect at their job, and they create viruses that don't have the DNA instructions, meaning they can't replicate further. Versus here, you have the one solid circles. Those are more successful particles, meaning they had the virus or the DNA packaged inside, so they can fully go on to probably infect other cells. All right, so now once we've collected our data set, you get thousands and thousands of these micrographs. And the rest of the work now is done inside computers using programs such as System. More popular now, there's also something called CryoSpark. So you have different options to process this data set of micrographs. And the first thing you do is the computer will pick out what is a particle so that you have those to work with. And then you'll take these particles and you'll classify them into different orientations of capsids known as 2D classification. And then the reconstruction is an iterative process, and the computer is full of refinements as well, taking all these different orientations in order to build this 3D model that you see here. So that's pretty cool. And then once you have that map or electron density, you're then able to take the actual protein structure, usually from the protein data bank, and try to fit that model into the map that you got from the cryo-EM data collection. And here's just to show that actually the higher the number is, the worse the resolution, and the smaller the number, the better your resolution is. Here's, you can see at 4 angstroms, it's a bit difficult to fit in all your side chains and amino acids. However, as you get better and better, you can see it's much clearer what the structure actually is and how to build your model. Yeah. So pretty much in our lab, we aim for around 2 angstrom to 3 angstrom is high resolution. Well, also excitingly, we've gone something under 2 angstroms, and in our lab, I have a lab mate who actually got 2 under 2 angstroms resolution structures this week, which is really exciting, because at that point, you can even see this thing we call a donut hole, which is this hole right here in the density. All right, so now that I've kind of given an introduction to the subject matter that we study and the technology we use to study parvoviruses, my project is looking at the family of Parvoviridae, which is the one that affects vertebrate hosts. And even though there's 10 genera, there are some that are really well characterized, such as Dependoparvovirus has a lot of structures because AAVs are used for gene therapy. However, there are actually 4 genera, as you can see here, Ave parvovirus, Copi, Ardi, and Lori parvovirus, that have absolutely no structures at all. And so my project is to kind of fill in the gaps that are missing within the subfamily of parvoviruses to better understand parvovirus distribution, diversity, and such. So these are the ones highlighted. And I'm using kind of my prior experience since I'm also highlighting ones that I've worked on in the past. So being somewhat familiar with parvoviruses to look into parvoviruses we know nothing about. And in this talk today, I'll be talking about recent work I've done to fill in the gap of Ave parvoviruses. Okay. And so just to kind of briefly refresh you on the flow through, we're looking at gaps in knowledge within the family Parvovirinae. We've been able to prepare the sample and send it for cryo-electron microscopy data collection. So getting 2D projections, getting the 2D classifications, alignment, 3D map, and then building the model. And then being able to get something like this to show how well our resolution is, to then build the monomer, and then build the entire capsid. All right, so 2 Ave parvoviruses that I've worked on in the recent times are actually, first one is red crowned crane parvovirus, which is an Ave parvovirus that was discovered fairly recently in 2018 from a metagenomic study using fecal samples from both domestic as well as wild red crowned cranes in China. And this is a really cool study because there's actually very few virological studies that have been done on this species. It's actually one of the rarest crane species in the world. However, the RCPV, or red crowned crane parvoviruses, actually, we don't know if they're pathogenic or not, so there still needs to be further studied. However, I think any studies on red crowned crane viruses would be good in order to help conservation efforts since they are a pretty rare species. On the other hand, another Ave parvovirus that I studied is the turkey parvovirus, and this one is actually pretty pathogenic. It was actually discovered in the 1980s. It affects primarily young juvenile turkeys with a whole bunch of terrible, terrible syndromes like runting stunting syndrome, poultry enteritis, and mortality syndrome. And there's currently no vaccine or drug to treat this. And generally, if to prevent a spread of this, you just have to cull many turkeys or chickens. So this one is actually a pretty cool one to try to study and see if any vaccine development could be furthered. All right, so I skipped the parts of preparing it, but I did actually make these viruses from sequence that I got from NCBI data bank. I cloned them and I made them in SF9 cells. And here you can see that I was able to make them successfully by the presence of particles in both of them that look like capsids that we saw earlier, as well as running an SDS gel to kind of make sure that the size of my sample was correct, which I expected to be about 60 kilodaltons, which you can see is about 60-ish. And then going through that entire workflow I mentioned earlier, I was able to get two structures, one for MAPS, one for RCPV and a map for TUPV at 2.66 angstrom and 2.35 angstrom, which are both pretty good. And here you can see that the maps show those characteristics I talked about earlier, which is the 5-fold channel here, the 2-fold depression here, as well as the 3-fold protrusion. And then here's just some cool details to look at the map further to see how good it is. And at 2.3 to 2.6 angstrom, you can see that the density, which is the black mesh, I can pretty successfully fit and identify where most of these amino acid side chains could go. And here I'm kind of just showing you examples of a region that's more conserved, so that beta barrel, as well as some of the variable loop regions, just to show kind of the resolution to model fit. And overall, I think the cryo-EM maps allowed the building of pretty reliable models. And here's another close-up from those models. I was able to build the VP, or the 1 out of 60 units that we're building the capsids. And from here, you can see it conserves this jelly roll motif again, which is those beta sheets that are kind of folded in the middle. And lastly, I wanted to compare, because there's no other structures of these out there that they haven't been published. I wanted to use something called AlphaFold 3, which is an AI that you can input any sequence and it'll try to predict what the structure would look like based on other published data. And since there were no other Ave parvoviruses published, I wanted to see how accurately my actual structure would compare to the predicted structure. And generally, AlphaFold 3 is pretty good. I think it was a 90 to 93% accuracy. Definitely, it got the conserved region based on all the parvovirus structures it learned on really well, here and here. And AlphaFold are in the blue colors, and the structure that I solved are in the red and brown colors. And really, any differences I noticed were more in the surface loops or variable regions, which is to be expected since that's what's the most different amongst all the parvoviruses. Right. So since I found AlphaFold 3 to be fairly reliable, I used it to kind of look at more Ave parvoviruses, since TUPV and RCPV are not the only viruses in this genre. And to make a point, so for Ave parvoviruses, all the members affected affect birds. So these are all bird viruses. And so here's my map that is cryo-EM determined of RCPV and TUPV. And the ones that we have not solved in this lab, used AlphaFold 3 to predict, are chicken parvovirus, pileated finch parvovirus, and pigeon parvovirus. I wish I included a picture here, but because the pileated finch is a cool bird and it has like a little red mohawk. Right? Okay, so lastly, because this is a new structure in a genre that recently had no structures, I wanted to compare it to kind of its other family members within the same subfamily. And kind of as expected, the conserved region is the same amongst all of them. And just to show that this is canine parvovirus, which is a protoparvovirus, and also caused that puppy parvo outbreak. Here you have Aleutian mink disease virus, human Boca virus, avian AAV, or now we're renaming it quail AAV, PAR4 and B19. These affect a wide range of hosts. And what is interesting to me is that in red is the RCPV that I solved, and it's much more truncated in these regions, which make up the 3-fold protrusion compared to other members of the same genre. So that could maybe be further analyzed and studied. Okay. And lastly, one of the experiments I did was to find a likely kind of glycan that it can recognize as a receptor, since that's really important for cell attachment. And so I did a cell binding assay using fluorescently labeled RCPVs. And through this experiment with FACS, or fluorescent activated cell sorting, I was able to determine that this glycan here was a potential receptor for RCPV, so that could provide some insights into maybe what type of cells it infects. Right, so pretty much the conclusions that we have is that we determined the first high-resolution structures for the Ave parvovirus genre, which is cool since there was no structures for this genre until now, as well as identifying a potential receptor for RCPV, could lead to further studies into its infectivity cycle and how it and which cells it identifies to infect. Future directions are I need to resolve more structures of the other 3 missing genera of parvovirinae to fill in the gaps of structural parvovirology, as well as investigate the basic biology. So look at receptors, look at mechanisms, and then that would provide further insights into this subfamily and family of parvoviruses as a whole. Right, thank you for listening. I'm open for questions, and here are the people I'd like to thank.
Thank you very much. You said, and once again, I have the microphone, so I get the first question. You said that you freeze these guys here in Gainesville and you ship them off somewhere. Where do you send them?
So that depends. So we have to look for time openings since these microscopes are servicing other structure labs as well. But recently we've sent some samples to Stanford for their microscope. At least for my samples, we actually sent them down to Scripps in Jupiter.
Oh, cool. So how do you keep them frozen while you're shipping them?
Pretty much you can use dry ice is the best way to keep samples frozen while shipping them. And normally when they're on the grid, it's easier to transport the grids to different microscopes.
Oh, they're in like liquid nitrogen or something?
I think dry ice is a bit easier
dry
to
ice.
ship.
Okay. I will stop now. There must be more questions. That was fascinating.
Thank you.
I even understood a lot of it.
Oh, that's good to
So
hear.
thank you very much. Engineers don't do much with these things.
Oh, yes.
So I really don't know very much about viruses. This was a great introduction. Other than AAV, that's the only one I know. So my question is this capsid that you're studying, when the virus is common to say jumps from chicken to humans or swine to humans, is it because there is a common capsid, common whatever you call this, the protein structure? What happens when you said some of these viruses only work, infect this species or that species or that, but the viruses that jump, do they mutate and jump, or are they just different viruses? And because of their protein capsid.
So what's really cool is viruses are constantly mutating and doing kind of a survival of the fittest. And I think it's still kind of hard to predict, but sometimes like in the case of canine parvovirus, just 5 or 6 changes might be enough for it to jump from one species to other. I know in case of flu or influenza, it was able to jump from chickens to human once it was mutated its surface enough that it recognized a receptor that's in our lungs, because initially it only recognized receptors that were more in the gastrointestinal, which is not as airborne. So because they're constantly mutating, sometimes just a few changes might make it fit enough to survive different environments and then recognize receptors that are in different hosts as well.
So it is these chains that mutate, right? The chains that you are showing of the protein.
Yes.
Right?
Proteins, yep. Oh, no problem.
Passing it around. You worked with the 2 genera, and then you said you grew them so that they could reproduce. And where did you get the cells, and do you actually grow them in your laboratory? I know how difficult that is, and you have to have a special
Incubator, yeah.
section and keep it. Do you grow them in your own lab?
Oh, yes. So for this one, actually, it was 2 viruses, but the same genre, so Ave parvovirus, but I'm working on other genre as well. So these were grown in something called SF9 cells or Spongiptera frugiptera cells, which came from marf larvae. and we grow them in flask. And there are two kind of cells that we have in the lab. We have also HEC293 or human embryonic kidney cells, which they stick to the bottom of a Petri plate so we can grow them in stacks. The SF9 cells that I used for this project actually are in a special incubator at 28 degrees Celsius, and they're put on a shaker since these cells don't like to stick to a surface and have to grow in suspension.
And I know you have to be
Yeah.
very careful because they might mutate and
Mhm.
go to people, so
Yep.
you have to address and so forth. Thank you.
That's true. Yep.
Well, thank you very much.
Thank you.
Once again, we've had some very interesting and very well done talks. Thank you for coming today. And I'll ask you, like I ask all the others,
Mhm.
maybe next year I will send an email to you and say, I need another speaker. Will you find one for me?
I think I can track a few down from my cohort.
Thank you very much. Okay.
Thank you.